Gene editing is one of the most powerful tools for studying gene function. But when cancer hides hundreds, maybe even thousands of potential disease-causing genes, how do we pinpoint the real culprit? Gene mutations and knockouts have helped us identify some well-known oncogenes, but there are still many more to discover. Moreover, traditional low-throughput research methods also make it really tough for us to pinpoint new functional targets in large-scale gene studies. To tackle this challenge, scientists developed CRISPR libraries. By combining gene editing with omics analysis, these libraries allows us to simultaneously and massively intervene in numerous genes within cell populations, pinpointing key tumor-driving targets. From basic research to industrial applications, CRISPR library screening is rewriting how we explore life sciences. Through diverse functional screenings, CRISPR screening is also showing endless potential across different research fields. In cancer drug resistance research, CRISPR screening can rapidly identify chemotherapy-resistant genes, accelerating the development of novel treatment strategies. Additionally, CRISPR library screening can also be used to identify host factors related to viral infections, shedding light on the keys to combating viruses. Meanwhile, it's making waves in antibody target screening, cellular signaling research, and non-coding gene function studies, with applications expanding far and wide. Also, with the development of in vivo screening, alongside integration with technologies like single-cell sequencing, CRISPR screening is now being applied to complex biological processes such as stem cell differentiation and aging. Ubigene offers efficient solutions to leading pharmaceutical companies and research institutions. Our mission? Make genome editing easier. We support every step, from gene discovery to clinical transformation.